Hi, so this is our lesson for causes of mutation. Mutations are changes to the genetic material, something that I taught you last lesson. And the word mutant refers to organisms or genes or chromosomes that carries those mutations and make them different from the wild type. The word wild type just means the normal population or the majority of the population. Now, how do you become a mutant? There are two ways. You can either inherit it from your ancestors, from your parents, or it can be acquired on your own. So your parents don't have them, but you have acquired them during your lifetime. So let's look at the first one, inherited mutations. Inherited mutations are being passed down the generation. If we have a look at these two diagrams, you can see that on this side, the red color means that there is mutation in that thing. And then on this side, if it's yellow, it's normal, but if it's red, it's mutated. So we look at the germline mutation, the red is a sperm and then the egg looks normal. That means the mutation actually came from the father's side. Now, if these two gametes, gamete is a word for egg and sperm, if these two fuses together and make one cell, and then that cell divides into many cells and create the human being, that means every single cell in the human is going to carry that genetic mutation. When that human is going to produce eggs or sperm to for the next generation, mostly half of the sperms or half of the eggs are going to also carry that mutation. Whereas in somatic mutation, the sperm and the eggs are completely normal. The embryo or the organism itself actually acquired it during their lifetime. So that usually happens randomly, or maybe something has caused it. They would only damage or mutate one cell's DNA. And so therefore, if that cell continued to develop, then the cells that are next to it and came from it will have that mutation. But it wouldn't affect the rest of the body because those were developed from normal, unmutated cells. And as a result, when this person produces sperms or produces eggs for the next generation, they're going to be normal because that part of the body that are involved in reproduction is completely normal and doesn't carry the mutation. There will only be one exclusion. That is, if this acquired mutation happened right at the reproductive organ that are involved in producing eggs and sperms, then you have a chance of having eggs and sperms that are affected. And that type of mutation then becomes germline mutation. So here's just a little summary of that. This is for your information only. You don't have to memorize any of these numbers or names. I just wanted to show you the amount or the percentage of cancer that's being inherited. You can see that ovarian cancer is a type of cancer with the highest inheritation rate. Stomach comes second, and then these are the rest of them. So for ovarian cancer, you can say that a lot of it is actually inherited. 20% is quite a lot. Dwarfism is another type of inherited genetic disorder that affects the skeletal muscular system's development. So you can see in this case, this is unaffected in dark blue, affected is in light blue. So the mother, in her genetics, she carries two normal alleles, so she's not affected. The father, however, carries one normal and one affected, and therefore he is affected. When the father produces his sperms, those sperms are going to have only half the number of chromosomes, meaning that these two chromosomes are being split up into two different sperms. So half of the father's sperms will contain the affected allele, whereas the other half will have the normal allele. Now, these two children in the middle are lucky because they inherited normal chromosome from the dad and a normal chromosome from the mom. Whereas these two on the sides in light blue, they have inherited the affected chromosome from the dad and a normal chromosome from the mom. So as a whole, he is affected, and she is also affected. Now, after talking about inherited mutation, we can talk about acquired mutation. So those are mutations that are not coming from your parents. You can't blame your parents for it. It's all on you. So there are also two smaller categories under acquired mutation. There is spontaneous, which means it just happens. We don't know the reason. Nothing has really caused it. And then we also have induced mutation, whereas the individual may have been exposed to some sort of mutation causing agents. So, acquired mutations, they're not inherited from the parents. If you have the spontaneous type, it means that it randomly happened without any causes. And where and when usually does that happen? Well, it happens during a process called DNA or RNA replication. You know by now that all cells have one copy of genetic material, and that is being kept inside the nucleus. And according to the cell theory, you know that all cells come from pre-existing cells, meaning that you have to divide your cells up into twos when you want to make a new cell. Now, when that happens, you can't just divide and chop up your chromosome and then chop up your cells like that. You have to first replicate your DNA. And this process of replicating DNA means that you're duplicating the genetic material. The end result is to have two cells with identical genetic material, identical DNA or identical RNA. This is what it means by duplicating. They have to be identical. Now, what has that got to do with mutations? Well, it turns out this process of DNA replication that happens inside the cycle of a cell's growth is the part where there's most amount of errors made. Because replicating DNA means that you have to play around with your DNA and try to find the matching base pairs and just making sure that it's identical. But this process of replicating it actually have a lot of errors that could spontaneously occur. And if that happens, mutation happens. Here's a diagram that shows you how fast things can mutate. We use viruses often as a model to study mutation. Sometimes we also use bacteria because they replicate really fast. So you can kind of speed up the life cycle and be able to see over time and over a certain number of generations, how much can I accumulate my mutations. In this diagram, you can see that at the bottom is the genome size. So 10 to the power of 7, that's large. 10 to the power of 2, that's smaller. So having a larger genome size means that you have more genes and more information. It could also indicate that you're a more complicated piece of machine. So you're a more complicated organism. Here on the left-hand side, mutation rate, 10 to the negative power of 2, 10 to the negative power of 10 is down here at the bottom. Remember, because this is a negative, so the larger the number, the smaller it is, all right? So here at the top, that's fast, a lot of mutation. At the bottom, not too much mutation. If we look at the red and the blue color, red means RNA and blue means DNA. Single-stranded and double-stranded is indicated by the abbreviations SS and DS. I do expect you to be able to interpret diagrams like this. I might have questions that ask you to interpret diagrams in the test. So if we look, we might be able to spot a few observations or trends or patterns. You can see that the reds are mostly on top and the blues are mostly at the bottom. Why is that? Well, turns out RNA, which is indicated by red, is a more primitive form of storing genetic information. An organism that uses RNA to store genetic information doesn't have an advanced proofreading system. So when they're replicating their DNA strands, they actually have less ways of proofreading their DNA and therefore, RNA, and therefore fixing any errors. Whereas viruses that uses DNA as the genetic material, they actually have the ability to proofread their resulting DNA from that replication process. And if they see any errors, they will get enzymes to go in and correct them so that those errors don't stay in the genetic material. And remember, the errors are the mutations. So more proofreading, more fixing means less genetic mutation. And so the mutation rate will be lower for these blue dots or crosses. So that's where spontaneous mutation could happen. And then now we're going to look at induced mutation. Those are mutations that are being caused by chemical or physical agents. Now, these agents, we call them mutagens, which are any factors or substances in the environment that could cause mutations. They cause mutation by maybe affecting the transcription process, or maybe affecting the DNA replication process, or they could kill your cell and force your other cells to do cell division and increase the chance of getting mutations during that cell division. They could impair or damage some of the functions of your particular genes. And over time, maybe sometimes having a small piece of mutation doesn't affect you, but if those are not corrected by enzymes or by the white blood cells, then you accumulate them over time, and it does, and usually negative effects come out of them. So there's three categories of these mutagens. You have radiation, you have chemicals, and you also have infectious agents. So let's look at the first one, radiation. If you remember what you learned in grade 9 science about energy, you would know that chemical bonds stores chemical energy, and energy can interfere with each other. So that means if you have another source of energy coming at your chemical bonds, you could break them and you can release some energy from those chemical bonds. Radiation itself is a form of energy, is pretty strong. So if it comes in and blasts at your DNA will actually get damaged because the chemical bonds inside DNA cannot hold itself together due to this exposure to energy. These pointing with the arrows are the many different types of bonds that you could potentially break in a piece of DNA. I'm not going to explain all of them in detail. That can be saved for university, so I don't need you to memorize what type of bond they're breaking, right? However, I do want you to understand that they break because of exposure to stronger energy, which is radiation. Irradiated food is a concept that a lot of people don't know about, but they should. So what we're referring to when we say irradiated food, we mean that we pass the food through a machine that can emit strong radiation at the food, and then those food is going to go into your supermarket and sit on your shelves. So this technology of irradiating food was invented in the States. You might wonder why? Because foods like onion and garlic, they are root. They're roots. So that means their root cells are going to sprout given the right condition. Your onion, naturally speaking, if you sit it on the shelf for 1 week, it might start it sprouting, or maybe 2 weeks. However, if you make it go through a radiation machine, the radiation will kill the cells or damage the cell's DNA to the point where it's too damaged to do any cell growth, and it would lose its normal function. So now you can have your onions sitting on your shelf for close to a month and they wouldn't sprout. You're extending the shelf life of your food by passing it through a radiation machine. A lot of supermarkets also do it to their meat, for example, their fish and chicken and steak, because if you pass it through those machines, they kill all the bacteria and the virus on it by emitting radiation at them. And then that also is going to stop it from going bad too fast. Now, they claim that this type of technology is only supposed to kill the cells or create enough mutation in the cells that are in the food. And the radiation itself doesn't stay with the food. And so people eating it should be safe. That's their claim. So I'll leave it up to you whether or not you want to believe that. Chemically induced. Oh, by the way, just want to say most food that you get in a normal supermarket, they've all been irradiated. They just didn't tell you because there's no way that they can let that sit there for 3 weeks for that long without having it go bad. So chemically induced, that's the next category of mutagens. Chemically induced mutations can be classified into a few categories. You can either have base analogs, or you can alter your base, or you can have something called an intercalating agent that changes your DNA. First one, base analog. So base analog is when you have a molecule that really look like your normal base. For example, thymine is a normal base in the DNA. Bromouracil is a very similar looking one. So if you look closely, you can see that the top right here is slightly different, but at first glance, they're almost identical. Now, when you're doing your DNA replication, what happened is that your bromouracil can come in and replace your thymines. However, after replacing it, bromouracil actually shows differences in base pairing from the original. So thymine would want to bond with an A, but bromouracil that replaces it wants to bond with a G instead. So is in that case, if you have this replacement happen, later on when you're doing cell replication or DNA replication, your bromocil is going to end up having different things bonding to it. And that's going to change the original sequence of your DNA. You get a substitution mutation. So here is an example of it. Bromouracil, instead of forming 2 bonds with an adenine, it is more than happy to form 3 bonds with a guanine or 3 bonds with a cytosine. And as a result, you get a change in the genetic sequence. The next category is the altering structure of a base. So instead of having a very similar looking one, you have even smaller molecules that could attach itself to a normal base, but after that, that base is going to be changed in terms of its bonding preferences. So here are 3 examples: hydroxylating agent or alkylating agents or deaminating agents down here. These are the diagrams. I'm just going to explain one of them to you and you can read the other ones on your own. Again, you don't need to memorize any of these chemicals, all right? I might show you diagrams in the test and ask you which type of agent you think this is, but I will never ask you to just come up with chemical names. So let's look at this example first. Cytosine is a normal base. It likes to form 3 bonds so that it can bond with a G who also likes 3 hydrogen bonds. Now, with a small little OH group, which is hydroxylation, attached to the cytosine, it's now only forming 2 bonds. And that would be either with an A, adenine, or it can be with a T. So you can see that it's supposed to bond with a G, but after this attachment, the bonding property has been altered. And therefore, the partner it would want to have is also going to be changed. Same with these other two. You can see that the pink is attached, and then that changes who they want to bond with. And so that's also going to cause trouble when you're replicating your DNA. The last category is what we call an intercalating agent. So those are flat multiple ring molecules that could kind of insert itself into a strand of DNA. After you insert it into the DNA, it's going to stretch it. And so the proofreading enzyme, when they're coming in and looking at the DNA, they're going to be fooled and think that, oh, there should be another base there. Let's put another one in. And you have created an insertion mutation. And insertion mutation, as you know it, it would cause frame shift. This is something that we talked about last class. So here's an example. This is your intercalating agent. It come and insert itself between the G and the A, creating extra space. The enzyme will come in, they will add another base here, and then you would have insertion mutation. And then the last category is kind of the catch-all category. Any chemical things that damages or changes your DNA, but does not fall into any of the previous 3 categories will come under this one. I'm not going to ask you too much about this category. Actually, I'm not going to ask you about this category in the test. You just need to know that it's a catch-all category. Okay, so we talked about radiation, we talked about chemicals, now we're going to look at infectious agents. Those are things like viruses and bacteria. They have the ability of inserting their own DNA into the host's DNA and changing the host cell into a factory machine that keeps on replicating the cell and spreading the viral DNA everywhere inside the host. Very typical example, human papilloma virus. So they are viruses that could inject their own DNA into the cervix membrane and turning those cervix cells into non-stop cell-dividing tumor cells. And so they become larger and larger, and that's cancerous. So this page kind of shows you some of the characteristics between cancer and non-cancer cells. I don't need you to memorize everything. I want you to read it and just pay attention to 2 of them. So number 1 is that the cell growth and cell division is completely out of control. It will just non-stop keep doing it. And the second one, the normal cell, it is much more controlled and much more systematic. It will only divide when there is a need to. The cell also doesn't communicate well with external cells, and it's disguising itself to immune cells. So your white blood cells and your immune cells has the ability to identify foreign material or foreign cells, or to identify mutated cells even. A lot of the times mutations are being picked up by white blood cells. And after picking up that mutation, they will go and kill the cell so that normal cells with normal DNA can do cell division and create some normal cells to replace it. However, cancer cells will also have mutations that changes the name tag on the external cell. So in Unit 2, we learned about membrane proteins, how proteins, some of them are called glycoprotein. They have little name tags on the protein that sits inside the membrane. And those name tags are going to wave to the white blood cells and tell them what type of cell I am and what is my health status. Cancer cells will try to mimic a normal cell's glycoprotein because glycoprotein is hugely dependent on the DNA of the cell. So if any changes happen in the DNA, it's going to be reflected by these. However, cancer cell has a very smart way of disguising themselves and try to appear normal to white blood cells so that they cannot be identified and killed. So that's about it. It's a lot of informations. And remember, the test is not about memorizing information. It's about being able to apply it and being able to see the relationship between different lessons. So it's a much higher level thinking than just memorizing things. So try and go back to your other lesson notes and see how those relate to each other. Some of the question may also be based on some of your understanding from previous units as well. That's going to be a small portion. So you have to have some basic understanding of previous units. All right, so I'm going to stop my video here. And I will not say happy studying. I know it's a lie for some people, but personally, I enjoy studying biology.